So we're now discussing the anatomy of the male reproductive system, and we'll get into a lot of the physiology and histology. Make sure when I am done with this chapter that I take the time and go back to this histology slide. That histology slide tells everything about the anatomy of the seminiferous tubule. So you can get this from the book. Okay, so the textbook has this, and I believe your lab manual also has this. And so it will help you in understanding where you find the various cells of spermatogenesis, because I won't go over what this is right now, but would like to go over this with you after we've discussed spermatogenesis and some of the other cells involved in this process. So now, going back to the last thing that we were talking about, which was the coverings of the testes. Remember, the tunica albuginea is the actual true testicular limit or covering, and then the tunica vaginalis actually comes from peritoneum, from the descent of the testes through the peritoneal wall out into the scrotum itself, because we have to have the testes at a lower temperature in order for normal spermatogenesis to occur. Spermatogenesis occurs in the seminiferous tubules. It states here in our notes, tightly coiled tubules that are the site of sperm cell production, which is technically called spermatogenesis. We will have 1 to 4 seminiferous tubules per lobule. And remember, a lobule is from the invagination of the tunica albuginea into the testicular structure. Reteat testes, that's what I want you to know. Reteat testes is this area right there. So collects the outflow from the seminiferous tubules. It's reticular, has a little lacy-like pattern to it. Then from the reteat testes, we have efferent ductules. So here are the efferent ductules, and that is the actual connection to the epididymis. So efferent, remember, means to leave, so efferent ductules. Then the next structure you need to know is the spermatic cord. The spermatic cord is that connective tissue sheath and the tunica vaginalis and all that encloses BV, blood vessels, nerves to the testes. And remember, the cremaster muscle is the muscle that actually is associated with the testes. The dartos is associated with the scrotal wall. And then the ductus deferens will all be inside that spermatic cord, which is there because all these structures are now external. So we need a little additional protection, support, and unit to help keep everything together. Let's take a look at the duct system outside of the testes. We got a sagittal in front of view for you guys here. And let's first just look at this and the various structures and things that are easily identifiable. B for bladder. The bladder has, at the base, has that trigone area. Remember, we have a single urethra and 2 ureters. And what I don't like, but they do this because I guess the graphics artist is making their own interpretation of what should be seen, is these ureters, to me, look like they're actually coming in a bit high. Remember, they come in at the base of the bladder, so these ureters look a little too high to me, but they're there to help you recognize them as ureters. So U for ureters. Going down here to the testes, we have the epididymis lying along, and this is actually true, lying along the posterior surface of the testes. And so the epididymis has a head, a body, and a tail. And the efferent ductules are connecting the epididymis to the testes itself. At the base of the epididymis is where the DD, ductus deferens, begins right here. So I want you to realize the ductus deferens actually lies even on the surface of the epididymis itself. So the ductus deferens is also called the vas deferens because of the surgical procedure called the vasectomy. But as we follow the ductus deferens, realize it's going to go through the inguinal canal. So it goes from external to internal, and they don't show it well here at all, but then makes a beeline for the ureters, the medial aspect of the ureters. So goes towards the ureters and then hooks on around the ureters And then we'll have a swelling. Our word for swelling is ampulla. Vestibule or ampulla. Vestibule is a big space. Ampulla is also a swelling. So the ductus deferens has a swelling, and it's called the ampulla. So when it's enlarged area, and at that area of the ampulla behind it, posterior to that, is a structure called a seminal vesicle. This is obviously bilateral. We're just looking at one side right now. So the ampulla and the seminal vesicles will come together, as you can see in this drawing, into what is called the ejaculatory duct. So the ejaculatory duct is in the prostate itself and will meet up with the prostatic urethra in the prostate. So then semen is actually deposited into the ejaculatory duct and into the prostatic urethra, and then we'll go through the rest of the urethra on its way out. And you can call it the penile urethra, but we call it the spongy urethra, and we'll see that in the notes. And we prefer to use that term as anatomists, and we'll explain what that actually means as we get more into that. There is an area of the urethra, really super small, that's called the membranous urethra. And that's because it goes through the pelvic diaphragm or muscular sling. In order to keep our organs from falling out, because you guys know at the bottom of the pelvis was a big hole, right? So we have a muscular sling right there to keep everything inside. And so this muscle that you see right here is part of that muscular sling to keep things inside. And in that area, let me erase it. In that area is a gland called the bulbourethral gland. And we have that also, oh, come on here. We have that also with the cat. You guys remember with your cat male anatomy, bulbourethral glands. That gland is really small, but is also responsible for producing a certain component of the ejaculate, and it's called the pre-ejaculate. We'll get into that in a little bit more detail. Let's take a look at a posterior view. Again, B for bladder, U for ureters, and again, we see head, body, tail of the epididymis, and you see the lobules that they have exposed in the testes itself. And here's the beginning of our DD, our ductus deferens, starts down there at the inferior pole of the epididymis or testes. And indeed, as we follow it on up, hooks on around again the ureters and makes a beeline for the prostate gland. Your cats, your male cats had the same thing. It's just your cat's prostate gland was not necessarily positioned at the base of the bladder. It was a little bit away from the base of the bladder. So the prostate gland, again, swelling ampulla of the ductus deferens, seminal vesicle coming together to form a short ejaculatory duct, which is not easy to demonstrate on this, but realize it will empty into the prostatic urethra. Then we'll go through the membranous urethra. It has it right there. I will erase this little arrow here, make it easier to see. Membranous urethra, very, very short segment. That's the shortest segment of the urethra. And then we have our spongy urethra listed down here, down this entire length. So that's called the spongy urethra. A little bit of some of the structure of the epididymis, ductus deferens, and prostate gland, and those structures you were not required to actually do histology on. But it goes to show you there's a lot of variability of the histology. So let's go ahead and make some notes now about the epididymis, ductus deferens, and the ejaculatory duct. The epididymis is the next part of the duct or tract, and it is interesting. In that little epididymis, we have 20 feet worth of coiled tube. So that's a lot of teeny tiny tube, right? That's a lot of teeny tiny tube that's actually coiled up, and it lies along the posterior side of the testes. And again, has head, body, and tail. And is connected to the testes at the area of the head by efferent ductules. So that's the connection there. Immature sperm are actually stored in the epididymis and they mature. The next part of the duct system that we will come along and follow is the ductus deferens. Nowhere near as long. It's 18 inches long and extends from the testis to the pelvic cavity and terminates in the prostate gland. It is designed to transport sperm from the epididymis into the short ejaculatory duct. Oh. Come on, you speller. So when I say it is designed to transport sperm from the epididymis into the short ejaculatory duct, what do you know about what has to be in the wall of this? Smooth muscle, because the smooth muscle is going to produce peristalsis to actually help with that transportation process. They aren't going to be wiggling that way. They are actually only going to go that way when peristalsis actually occurs in the ductus deferens. So that's an important aspect to realize about that. And it gives it structure. The ductus deferens is a palpable structure, and that's why vasectomies can be done as an outpatient surgery, because you can actually feel the ductus deferens through the scrotum. You can feel the spermatic cord, and you can actually figure out by palpation or feel which one is actually the ductus deferens, because it has structure to it. It is solid. And so vasectomies can be done by making a little small cut into the scrotal sac, grabbing onto the spermatic cord, and then making a very small incision into the spermatic cord. And easily, by palpation, without feel, you can actually get the ductus deferens. You can pull it on out, and then what you do is you go tie, snip, and put it back and let go back in. And then not much of a tie for the outer aspect because your incision is so small. It is that easy to actually feel or palpate. So it has true structure. It really does. The ejaculatory duct, short, 1 to 2 centimeters long, and has a really interesting lining, pseudostratified ciliated columnar lining. It is the connection between the ductus deferens. Oh, well, actually, it is not the connection between the ductus deferens and seminal vesicles. It is where the ductus deferens and seminal vesicles come together, connect in the prostate to actually then deposit a spermatozoa into the prostatic urethra. And so we want to make sure that you remember the urethra in the male has 3 regions or divisions. We have the prostatic urethra going through the prostate, the membranous urethra going through the urogenital diaphragm, and then the spongy urethra going through the penis. Now, let's take a look at the product of ejaculation, which is called semen. So it is an accumulation of different material from different areas. It's going to be sperm cells from the testes that made its way into the epididymis and then the ductus deferens and therefore into the ejaculatory duct and into the prostatic urethra. But then we have other material actually producing the bulk of the semen. Seminal vesicles and the prostate are going to actually contribute to the bulk of that product or material that we know as semen. Our notes describe seminal vesicles as being on the posterior wall of the bladder. It is adjacent to the ampulla of the ductus deferens and produces 70% of semen volume. It has an alkaline pH. The secretions from the seminal vesicles will empty into the ejaculatory duct to mix with the sperm. What's interesting about the seminal vesicles, it has a dual role. It has to provide nutrition for the sperm cells, also has to provide a mechanism to help them further their maturational process to where they are capable of fertilizing an egg. So there's a difference. Sperm cells are not automatically capable of fertilizing an egg when they're formed. So that's something that you may not have known. Sperm cells have to undergo a grooming process in order to become capable of actually fertilizing an egg. So, and that is actually transportation through the male reproductive system and transportation through the female reproductive system. All produces a process that we're going to learn about in the next chapter called capacitation. It makes them capable of fertilization. So capacitation kind of improves their capability. So sperm cells are not automatically capable of fertilizing an egg when they come into contact with that. So what the seminal vesicle fluid, it actually provides nutrients for our sperm cells. And the main nutrient that it provides for sperm cell is fructose. Also, we will have prostaglandins in this seminal vesicle fluid, and the prostaglandins help somewhat with cervical dilation. So that cervical canal is ordinarily nice and tight, difficult to traverse, and so the prostaglandins will actually help to soften that up and to make it more capable of allowing sperm cell entry. So that's an important component of semen, that material from seminal vesicles. Now the prostate, we know, is just below the urinary bladder and encloses the prostatic urethra and makes up to about 70% of semen volume. So the bulk of semen volume is actually from these two glands. So approximately 30% of semen volume. 70% from seminal vesicles and almost 30% from the prostate gland. What's interesting, this is actually slightly acidic. And has citric acid, which is also a nutrient. For the sperm cells. The prostate gland also produces a very specific chemical called PSA. Produces prostatic specific antigen, PSA. This helps to liquefy semen after ejaculation. So in other words, very viscous material and harder to escape this viscous environment. So PSA will actually liquefy it, allowing the sperm cells to actually migrate along the reproductive system of the female, if that's where it's actually being deposited. Now, the secretions from the seminal vesicles as well as the ductus deferens enter into the urinary system here at the prostate. So secretions from DD, ductus deferens, and SV, seminal vesicles. And so those secretions or products will enter there, mix with the citric acid as well as PSA, and all of that is going to go together to combine semen with some of the secretions from the bulbourethral glands, which is actually inferior to the prostate. So it's in the membranous urethra, not in the prostatic urethra. So the next part of the urethra is the membranous urethra, and slightly lateral to this. The bulbourethral glands produce the pre-ejaculate. The pre-ejaculate happens to be slightly alkaline. And this is important because this viscous material is going to help neutralize the acidic urethra, the spongy urethra, because urine typically has an acidic aspect to it. So it's going to help neutralize that and also to help lubricate the spongy urethra for the ejaculate. So slightly alkaline, viscous, and is able to lubricate. Not lubricate. Jeez. Some days. Spongy urethra. Then the anatomy of the penis. The external genitalia is going to be the scrotum and the penis. And what we're going to do here is realize with the penis we have an area that's attached to the body. It's called the root or the creux of the penis. And then we'll have, with that, we're going to have a free shaft, also known as the body. And then the enlarged tip, which is called the glans penis. The penis is, all of that is basically erectile tissues because of the vascularity. So we see here vascular spaces, and we have 3 columns of vascular spaces. So this is very vascular tissue. We have 1 corpus spongiosum. Corpus, like nucleus. Corpus spongiosum, that's singular. And we have 2 corpora cavernosa. The corpus spongiosum is why we call that part of the urethra the spongy urethra. That is surrounding the urethra itself and indeed expands distally to actually form the glans penis, because that's basically the glans penis is part of the corpus spongiosum. The corpora cavernosa, paired columns dorsal to the spongiosum, and are considered to be the major erectile tissues. And the process of erection involves vascular engorgement. Remember, with the female, there's vascular engorgement that occurs, and this is a parasympathetic dominant process. So the blood vessels to the genitalia are under parasympathetic control. That will cause vascular dilation. So the arteries are going to dilate, going to the spongiosum and the cavernosa. So the arteries feeding into those vascular spaces are going to dilate, filling them up. Just like a sponge, I want you to realize that the structure inside is like a sponge that you actually use in household cleaning. And so when you fill it up, remember it gets very firm when it's filled up with water. And so we're going to fill it up with blood, arterial blood. The arterial blood, what's important about this is, do you guys remember how I told you we don't have reflux into our ureters? Do you guys remember what was responsible for stopping reflux? The weight of the bladder. As you fill the bladder up, it gets heavier and it pushes down on those ureters. Remember, the ureters are at the base of the bladder. That's why I said I don't like those diagrams, because they really should be down there at the base of the bladder. So as the bladder fills up, it becomes heavier and is a better structure to add weight to keep the openings of the ureters to the bladder closed. And that's another reason why we have to have peristalsis, because as the bladder fills, we have to literally squeeze that urine into the bladder itself. So the same process is here. As the erectile tissues of the penis fills up with blood, the weight of them actually cuts off venous outflow. And so that is what causes the erection or the firmness that occurs during an erection. And so when men have erectile dysfunction, ED, what do you know about the ability to actually cause blood flow to not leave the penis? That's the problem. So they're not having enough vascular fill or engorgement to weigh down and close off the venous outflow. That's how an erection occurs. So if you have these vascular tissues that are filling up and making themselves so heavy to make the penis firm and for penetration, you have to realize that this guy, the urethra, is at risk. It's at risk for doing what if you're filling up and making things weighty? It's at risk for collapse, correct? That's what the spongy urethra is all about. The spongy urethra is actually designed to do the opposite of allowing collapse. As it fills up, it keeps the opening patent. And see, that patency is what allows the ejaculate to actually leave. And so that's the mechanism that's very important. We need to actually dilate the arteries and arterioles feeding the penis and make sure the blood flow goes in and starts to compress the outflow, which is venous, and that will allow for an erection to actually occur. So that's that mechanism. ED, erectile dysfunction, has medical treatments, and the medical treatments actually allow for more efficient or a better inflow, which will then help to minimize the outflow because of the weight of the engorgement that actually occurs. Yes?
ED, is it because the arteries aren't getting enough blood there, or is it because the venous side is draining too much? Or is it a combo?
You're not, with this, you're not getting enough fill, an adequate fill. And so our parasympathetic system is responsible for this, and so we're not getting enough parasympathetic innervation to cause this, and so these medications improve the ability for parasympathetic vascular dilation to actually occur.
So would you say the spongy urethra is actually pulling?
The spongy urethra
Yeah.
will allow,
Keeping that.
as the weight fills and pulls down, the spongy urethra says, no, we're not going to block off this. So it actually does the opposite of allowing collapse. It causes more expansion or opening, keeping the lumen patent. So, but it is a vascular space too, but it does the opposite. It doesn't collapse. As it fills, make sure that the urethra stays open. Otherwise, you'd have retrograde ejaculation, which is not going to be beneficial for anybody. Okay, semen is a mixture of the stuff that we talked about earlier, a mixture of spermatozoa, which is sperm cells, and secretions from the accessory glands. Ultimately, it is an alkaline fluid. So any of the acid that we actually added didn't do enough to actually decrease the pH. Alkaline fluid with nutrients and chemicals to activate and protect the cells and activation is part of that process of making them capable of actually fertilizing an egg or penetrating the egg. Typical volume of ejaculate is anywhere from 2 to 5 mLs or cc's, whatever you prefer, which means we'll have anywhere from 200 to 500 million sperm cells per ejaculate. What's the population of the United States? It's a little over 300 million. So that's a lot of sperm cells. And I told you earlier that only a very few, compared to what actually is in an ejaculate, actually makes it to the egg for fertilization. So only 100 or 200 of the sperm cells that are deposited into the female reproductive tract actually make it to the egg for fertilization. And it isn't going to be the early bird that catches the worm. It's actually the latecomer that will actually be the one to actually fertilize the egg. And we'll discuss that when we go into more detail on that chapter. Anything else? Okay, spermatogenesis, like oogenesis. By the way, one of the students found a really good video about oogenesis and posted it into the 228 form, and I highly recommend that you actually listen to it. They said that as far as oogonia forming and primary oocytes, they said just one or a few, and it's actually more than just one or a few. We actually recruit a little group, a little cluster of oocytes to actually start the oogenesis process or complete it. With spermatogenesis, like oogenesis, formation of the gamete, which is for the male is the sperm cells, and that is a meiotic process. So remember, meiosis is cell division that is only done by our reproductive cells, because meiosis will decrease our chromosome number in half to form the gametes. So go from 46 to 23 for our gametes, male and female. That's what meiosis is all about. Meiosis 1 is reduction division. Chromosomes replicate, cell divides. We go from diploid to haploid. Then in meiosis 2, that's the equatorial division. This follows meiosis 1. No chromoreplication. We're stuck with chromosomes that we replicated in meiosis 1. No chromoreplication. just cell division to give us our actual haploid gametes. And we will end up with OR. Remember how many gametes, the final gametes from the female system? 1. Those polar bodies, not worth anything. We want one big cell. In this process, big isn't important. Motility is important, and that's when we will discuss spermiogenesis. So what we will do with spermiogenesis is different from spermatogenesis. We'll talk about that afterwards. But we're going to stop here, but before we do, I want you to know that this cell here, spermatogonium, is mitotically active. So remember, with mitosis, we get an exact 2 daughter cells that are the exact same copy of the mother cell. So oogonium, spermatogonium, same thing. 46 chromosomes, mitotically active. That is where everything comes from those guys. Difference in oogonium for the female, once a female is born, she doesn't have any oogonium. She only has primary oocytes. With men, they have spermatogonium their entire life. And this is one reason why women have a limited time of fertility and men have fertility actually beyond puberty. So we'll stop there and do another clicker question.